All right. Good morning, Canada. This is amazing. We have 45 people and counting coming in. We had 70 pre-registered for this, so we're very excited to have you on. Mike Kasten, who works with me at Finger Lakes Institute, will be letting people into the meeting, and he's going to be just kind of keeping track of questions. Housekeeping things, please mute yourselves. A lot of us are working at home still, or just at home, and there's fire engines and dogs and all those good things that you'll all hear. So mute yourselves if you can. If not, Mike will end up muting you too for you. Also, questions. We got, as I said, we're 45 people or better. We're going to too many people to have an open discussion. So if you have questions, please use the chat box. Usually at the bottom of your screen. Mike will be reading through those and making mental notes. And I hope to stop at a couple times through the morning here to take one or two questions each, and then we'll get to all the questions at the end. So with that, I hope everybody can see my screen. Somebody give me Mike. Will you go to my screen? Can you see?
Yeah, we can see your screen.
Okay. Fantastic.
It's a bit blurry, but
because it's a small one, it's been blown up too much.
Oh, should I make it smaller?
No, that's fine. We'll change it.
All right, we'll get started. Well, you all know by now, my name is David Carr. I'm with the Finger Lakes Institute, and I manage the Starry Stonework Collaborative for the Great Lakes. And we're going to talk about a variety of things today. I hope to get a little agenda coming up, and I hope to keep this going relatively quickly, not overwhelm you. And then at the end, Brick Schreier from EDDMapS is going to give an actual training session on how to use EDDMapS. And we're keeping that to the end. So if you're already familiar with it, as some people are, you can jump off and skip that part. But we're hoping we'll get out of here before lunchtime for sure. So first thing I'm going to have you do is look at this and write it down if you can. I will follow up with an email. This is the only thing I promise you have to write down. This is let's see here, where's my thing? This is a group of documents. Let me pull it up here. Here we go. It should look like this when you go there. There's lots of information in here. This number one, links to references and contacts. I'm going to try and bring that up live here. Okay, let's try and, okay. This is a bunch of links, a lot of information. This document you could download and email to somebody, and they would actually, if they wanted to get back to here, they could do that right through this document. So you get right back here again. Okay, we're back to this again. So again, this location has a whole bunch of very useful documents. Let me bring that back up again. Okay, here's this one document in particular is the first one. It's 01. There's lots of information here. And then there's a variety of other things here. If I can bring this back up, our video glitch kind of messed me up here.
Could you put that link in the chat?
Yep, I'm going to send an email out afterwards too, but I will, yes. So this is what it looks like. There's that first one. This folder here, there's all kinds of fact sheets and information about invasive species. There's Curly-leaf pondweed, Eurasian water milfoil, hydrilla, of course, starry stonewort and water chestnut. And there's at least two identification fact sheets and things in each one of those. These first two or three describe the collaborative. There's some information about our own New York State Department of Environmental Conservation and some other research that have done, again, more information about how to identify invasive species, aquatic plants. We threw something in here about harmful algal blooms, which are a problem down here. I know that's not our focus, but it's there. And then this actual presentation I'm giving today in PDF format is here as well. So we got lots of information available for you. Okay. So briefly, I'm going to talk about the collaborative itself. Just a quick background, who we are and what we're doing. We're going to talk about what an invasive species is. And I know a lot of you folks are aware of all this, but we're going to go through and do some of the basics anyway. A little repetition never hurts. We're going to talk about how to do a survey. And the survey means going out and collecting information or data from the natural environment in this case. And, the survey and sampling steps, how you're going to do it. We're going to use the term rake toss a lot, and we're going to have a little video that a coworker did about how to do rake tossing. And then we'll get into the hardcore stuff of identifying invasive species. And Tyler Harrow-Lyle is on with us today. He's from Ontario Technical University, and he's going to talk a little bit about that too. I have a little video about identification from a researcher in Wisconsin. So we're going to hit you with lots of information there. And we'll have questions in between. I can only take, we have time, we'll do 1 or 2 questions between each section. And at some point we'll have a stretch, 5 minutes, get up, walk around, get more coffee kind of thing. And then at the end, again, we're going to have Brooke Schreier going to do some training on EDDMapS for about 25 minutes. And more questions at the end of that. So quickly getting into the collaborative itself. We're located at the Finger Lakes Institute, which is at Hobart and William Smith Colleges in Geneva, New York, right in the center of the Finger Lakes region. It's a beautiful region. The Finger Lakes Institute, as it says there, promote environmental research and education in the Finger Lakes and surrounding environments. And then with this project, way outside the surrounding environment, because we're all across the Great Lakes Basin. And we have a variety of program areas: research, education, outreach, economic development. Hosted at the Finger Lakes Institute is the Finger Lakes Partnership for Regional Invasive Species Management. It's a mouthful, so we just call it Finger Lakes PRISM. There are 8 PRISMs across New York State. It's a really neat structure. All of them do the same thing. They fight and partner with anybody, bring in local organizations, state organizations, whatever it is, to as it says there, reduce the introduction, spread, and impact of invasive species. And we do lots of education, outreach, actually get out in the field ourselves, do prevention control methods, whatever we can to fight invasive species, both aquatic and terrestrial. So we have 8 of those across the state, and it's a pretty neat structure. So real quick to put us on the map, so to speak, this is Lake Ontario, the eastern end of Lake Ontario. We're right here in the center of the Finger Lakes region. For scale, this lake, this is Seneca Lake, and this is Cayuga Lake. These are each about 38 miles long. The big lakes, as you can see, they're all kind of long and skinny, hence the term the Finger Lakes. So now that COVID is passing, come find us, come see us. It's a beautiful area. Particularly in the summertime. Winters are rough, I'm sure, similar to what you have with the influence from all the Great Lakes, but it's a real pretty area. So the Starry Summer Collaborative, this is a Great Lakes Basin project. Okay, we went way outside what we're used to, working at the Finger Lakes region, and I'll have a map in a second of what that looks like. Our whole goal was to enhance the capacity of experts, resource managers, local stakeholders such as yourselves to address starry stonewort. We looked at ecology, outreach, and control. Ecology, we've basically left to the experts as best we can and report their findings. We've done worked hard at trying to get distribute information. There's a library, we call it, on the Starry Stonewort website, where there's a lot of professional papers and things up there. Outreach efforts, we do a lot ourselves. And of course, this citizen science program is the centerpiece for that, trying to raise awareness and get more folks out there learning about invasive species and how to learn how to manage them. And then control techniques, that's an interesting situation where it says BMPs there for best management practices. Our goal is to get to best management practices for starry stonewort, but as of yet, there really aren't any. And the big bad news I'll give you right off the bat here is that there's no known control strategy or method to eradicate starry stonewort. Hand pulling and diversicious suction harvesting shows some promise in small localized areas that done properly. But really there's no magic bullet at this point to get rid of this stuff. You can knock it down with algicides and things, but it's just going to come back. So we're working toward hopefully someday having best management practices, but we will be, Mike is actually working hard on a little bit of a management plan that will kind of overview many of the different techniques that are being used at the moment in research to control this stuff at least. Very quickly, at the bottom is the Finger Lakes Institute. This little diagram I came up with. My desk is way down at the bottom point there. Above us, we have an expert panel, and these are professionals in the industry, if we'll call it an industry. They're academics and researchers. We have about 8 of them. We talk to them on the phone a lot, and they're kind of our advisors. And then above them, we have collaborators. We've got 30 to 40 of them across the Great Lakes Basin. And these are people that are, again, academics, state agencies, non-government organizations, all working toward trying to control starry stonewort and other invasives, and also to help us reach out to the most important level, which is you folks, our citizen scientist group. And your group is growing the fastest. We've had some great growth in New York State too. I think we have nearly 100 people involved in our citizen scientists here in New York. Last year we had a small group in Indiana, and then we have a lot of you folks up there, and it's really great to see, and I'm going to talk about that. So that's kind of how we're structured, the whole thing. And the collaborator model was set up so, being remote to a very large region, we asked the collaborators to help us reach out and find, contact the citizen scientists and be our go-through. And again, I said these are advisors, the expert panelists, but the collaborators have been very helpful too, very supportive. So here's the map. Okay, that green line, that is the Great Lakes Basin. I could international there. I'm all excited about that. This is a big project. Put a little star there in your region. This is the Kawartha Lakes region. Most of you on this call are from Kawartha Lakes. I think there's some others, hopefully from a couple of locations in Canada. And then this little thing, again, that's where we are down here. So it's a very large area, but it's kind of exciting. So that's some very basics about the collaborative. Where are we going in the future? This is a good news, bad news slide. I'm going to pick on Carol Cole. She's my friend there at the Kawartha Lake Stewards group. She knows this. A few others know this too. Our grant funding is running out here in September. So I'm hitting you with the bad news right up front at the beginning of the session here. However, we're not just disappearing. We're going to keep as many of our resources up online for as long as we can, at least a year. Our website's got a wealth of information. Again, there's a little library on the website you can go to. The resource I showed you at the very beginning that you can access, I'm going to keep that up for as long as possible and so on. So we're not going to completely disappear. And we are always looking for funding opportunities and ways to rejuvenate things and keep it going. But the good news is you folks in Canada have done just an amazing job. I mean, there's so much momentum in Southern Ontario in such a short time. I mean, the collaborative has been a collaborative for 3 to 4 years. I've been involved in about 3 years. And in less time, in about 2 years, there's a whole network of people in Southern Ontario that are really excited and really looking at how to control starry stonewort. And I just want to show you that. I came up with this little graphic. There's this network that has blossomed. And just as my cheat sheet here, because I'll get names wrong if I do this, I don't do this right. All these stars, the big star in the middle again is roughly the Kawartha Lakes region. So we got Kawartha Lake Stewards and Kawartha Conservation. And thanks again to Carol Cole and Brent Trigano, he up there at Kawartha Conservation. Carol stepped in early from citizen scientist and jumped up to collaborator in my mind. She's really helped get a lot of things organized up there. I know she's on this call. Thank you, Carol. Get things organized, and then Brett stepped up and got his group involved. So, that's why we have, I'm looking here, 56 people on this call, and others that wanted to be on and couldn't make it and are still going to be involved. So it's pretty exciting. It's a big bunch of people in one area to really get involved in this program. The Severn Sound Environmental Association, I don't know if Tamara's on, I hope she is. She got involved, and so we've got people all over Georgia
I'm
Bay
here.
now. Yay, thank you for being on, Tamara. So they're involved now. They want to learn about starry stonewort and get their folks involved in doing, you know, monitoring and so on, and control methods. Over here, we got the Nature Conservancy of Canada, Amanda Tracy. I'm not sure she's on today, but she was going to hopefully get a couple of their interns involved. She's been very supportive of what we've done. I gave them training last year. Unfortunately, COVID is hitting that agency, you know, with some difficulties, as all of us, but they've been very supportive. And you know, Nature Conservancy is a great worldwide organization, does great things. So it's nice to have them involved. The Ontario Federation of Anglers and Hunters and EDDMapS is with us, or will be. That's Brooke Schreier. He'll be on later. Again, he's the main contact for EDDMapS in Ontario. And again, it's great to have him with us and working towards the common goal of trying to learn how to control starry stonewort. Lake Simcoe Regional Conservation Authority. Dr. Brian Ginn has been incredibly supportive through the last 2 or 3 years. He's done some presentations and webinars for us, done some great research. He's a limnologist with them, great source of information and help. And then Ontario Tech University, Tyler Harrell-Lyle, as I said, is on. He works with Dr. Andrea Kirkwood. They're doing fantastic research all through that region, looking at and sampling, taking, you know, samples of starry stonewort, looking at what it does and how it does it and so on. And Tyler gave us a neat webinar not too long ago, which is archived now up on our website. You can watch that. So here's this network, and it's not only lots of people of, you know, citizen scientists, but professionals, academics, and so on. This is amazing. I mean, you folks are doing just a tremendous job, and it's growing very rapidly. And many of you on the call or on this little diagram have already talked to each other via email or even a Zoom call. Several months ago, for example, there was a meeting that I did not attend, and there was 30 of you, which involved, I think, most everybody on this little map, if maybe not Severn Sound yet, because I hadn't heard from Tamara at that point. But 30 of you talking, you know, with Andrea Kirkwood and Brian Ginn and all those folks, Nature Conservancy, talking about what to do about starry stonewort. So that's just amazing. You've already built a wonderful network. We're just growing even further with this session. And beyond, I know Carol and some others have talked with some of the researchers in states of Minnesota, Wisconsin, maybe Michigan. There's some tremendous research going on in those. So that network extends that way. And I've been lucky to be able to talk with those people and make some connections for you. But there's no reason you can't keep the connections running. So that's for me, I think this is a huge pat on the back for all you folks that you're just running with this. I know it's a, you know, when everybody has a common enemy, you know, being starry stonewort, you jump on things, but you've just done a great job up there. So let's just jump right in. I want to talk about what invasive species actually are just for a minute. I mean, a lot of you know this already. We'll just go through it quickly. They are non-native to an ecosystem, and they can cause economic, environmental, and harm to human health as well, depending what it is. This is an overall definition, so I'm not talking about just starry stonewort. I'm talking about terrestrials, insects, bugs, animals, everything, all invasive species. They cause economic impacts, you know, agriculture, recreation, wood forest, et cetera, et cetera. You know, when we're talking about starry stonewort and other invasives, they clog the waterways potentially. It impacts, you know, tourism, boating and swimming and fishing. All those things are made very difficult because it has an economic impact. Environmentally, they push everything else out. They impact biodiversity, natural processes, aesthetics, all those things that, bad things that an aquatic or any invasive does, and human health. In some instances, there's direct human health impacts to from invasives, which is a problem. So some more information. Invasive species generally don't have any natural enemies. They can reproduce quickly. They adapt very well, even in the harshest conditions. As I said, they affect biodiversity and ecosystems. They threaten native species, and push them out. And so we have the, you know, the silly sci-fi picture here, but it's the same thing. You know, you have multiple invasive species, and there's some studies that, you know, maybe some help others inadvertently. There's some research I know. I think Tyler said that some invasive mollusks will attach to starry stonewort and things like that. So not good. They're a problem, right? This is probably the most technical thing I have on here, but it's important. This is what we call the invasion curve. I know there's a bunch of you here from last year, and thank you for coming back and helping us again. You may have seen this already. Across the bottom, time increases to the right. As that happens, control costs and areas infested increase, going up. At the very left hand, we have prevention. That's the most important place to be. We keep the things out to begin with. Keep the invasive species away from your water or wherever your area is in the first place. And that's where it's this ideal time for intervention kind of spans there. I don't know if you see my cursor from here to here through the beginnings of eradication. This is the best place to get involved and take care of these things. This is where they're introduced. At the end of prevention, we get the introduction of invasive species. Eradication is generally possible. Okay, I'm talking about generalities of all invasive species. In this first block, eradication may be possible, but as it climbs here, and again, as the areas infested increase, containment becomes more likely as far as not being able to eradicate it. Maybe we can contain it and keep it controlled to some degree. Unfortunately, this is where public awareness typically begins. And we don't want that. So your job here is not only to learn how to survey for starry stonewort and other invasives, but also spread the word. Okay, spread the word, educate your friends and family, people. We don't want to know, we want to be way back here at prevention. Okay, this is where we want to be. We're typical. We want public awareness to understand we need to prevent these things, not wait till it's already happening and becoming a problem. I have early detection here, which is kind of in the not very clear, and we'll talk about that in a minute too. That's important. Early detection is finding this stuff as early as we can. And then you get up in this disaster, and these are areas that we've all seen, various invasive species. There's not much we can do except manage it best we can, try and protect the existing resources and so on. And the costs go up exponentially for all these things. The further down the road you get with more infestation of invasive species, the higher the control costs get. So down to the bottom here, early detection, rapid response. Hang on a sec, mess them up. Okay, early detection, rapid response. That's a phrase we use all the time, because the earlier you find it and detect it, there might be a chance to rapidly get in there and do something about it, be it chemical treatment or getting a bunch of volunteers in to find out where it is and map it, which is essentially what you folks are going to do. And so on. So again, these are a variety of posters and flyers and things that have been put out by a variety of people, including us. And the whole idea is, again, get the word out there. We don't want these things. Clean your boat off, clean your RV off in a safe place, and dispose of the weeds or plants or whatever it is before you spread them to another lake or another area. So prevention equals protection, which means money saved. And of course, protecting our favorite environment and natural resources. Questions at this point? I'll take a couple. If there's just a couple questions, or we can just move on. I seem to have lost, hang on a second. Nope, sorry folks. Can you all see me still? Mm-hmm. Okay, thank you. I had a second feed of all your faces on another monitor, just disappeared, but I'll take it for granted you guys are still with me. Okay, I see some conversation going on in the chat box, fantastic. Okay, so I'm gonna move on and get into the survey steps here. We're doing great on time. I don't want to keep you guys all day for sure. I want to get you out before lunch. So this is the survey steps. I'm gonna talk about, this is actually what you're gonna do after this meeting. You'll be hopefully raring to go and get out there and do what we gotta do. Okay, quickly, what is a citizen scientist? We sometimes use the term community scientist. Same thing, basically. So it's defined loosely as scientific work undertaken by members of the general public, often, collaborating with or under the direction of professional scientists in scientific institutions. And again, as I said in that map before, the network you guys have built, you have the backbone. You've got the university, and you've got Brian Ginnick at your disposal and things, and so there's a lot of support there. So keep building those relationships. The goal of the survey, other than, as I said, awareness and understanding, the importance of invasive species and why we need to control them, is we're going to ask you folks to identify between 1 and 5 high priority invasive species. Now, these are suggestions. If you are only comfortable trying to identify stars, don't worry, that's fine. But we figure you're already throwing a rake out there, maybe try and identify some other other invasive species like Eurasian water milfoil and the others listed there. Because it's all helpful data, it's all useful, it all get loaded into EDDMapS, and it goes into the major master database, and it's all very valuable information. So if you can, identify more than just one, but we're not gonna hold this over your head and come up there and take a trip to Canada and beat you or something to do more. But as much as you want to do is great. If you want to get into this, we got lots of resources available for you. Just learn how to identify them. But try and identify, 2 or 3 at least, if you can. So here it is. This is in a nutshell what we're going to ask you to do. I call it the protocol, a big fancy word. We're going to ask you to do rake tosses in 1 to 3 locations. Okay, this can be from a boat, kayak, canoe, off your dock, if you've got lakefront property, off your shoreline, whatever works. We're only going to ask you to do this every other week through the summer, July through October. So that works out every other week. That works out to 8 weeks. If you do 3 rake tosses, that's 24 rake tosses, and for the whole summer. That's not a lot of work. We want to make this easy for you, but it's important. Once you've done the rake toss, and again, I've got a video I'm going to try and show here in a minute that describes how to do it. You look at what you found, and you're going to record it in EDDMapS, and I'll talk about that in a minute too. I got to say the safety first. I know most of us love to go out in the water by ourselves or on the water. It's calming and peaceful and things, but be careful. Take a buddy with you. Go in groups, and groups are fun because you can share information. And both, if you're new to identifying aquatic plants, you can kind of bounce it off each other with a field guide or something, and figure out what's best and what you're identifying. And then check in. That's my thing. I'm going to try and send up periodic emails to the group, just to maybe throw out some information that's pertinent to what you're doing, and checking to see how you're doing and so on, how it's going. With Brooke's help, I will be kind of monitoring what you're logging in EDDMapS, so you can tell how you're doing. Of course, you can send me questions. If all 50 people like today send me 3 questions, it would be a little hard to get back to you on all of them, but go ahead and send questions if you need to. Keep it simple, okay? Again, we're trying to raise awareness, but get more boots on the ground. This is not just, it is for you to raise awareness for you and learn what's going on out there as far as invasive species, but the data you collect is valuable. So we're keeping it simple with 1 to 3 locations every other week. That's not much, but if you look at this, if you do that 24 rake tosses, if 50 people do it, that's 1,200 rake tosses for the summer. That's suddenly a really big chunk of data. That's very valuable information. There's a lot there. Strength in numbers, but keeping it simple at the same time. Now, if you want to do more than that, please feel free. If you want to go out every Tuesday afternoon at 3 and do your rake tosses, that's fine. It's okay to do more than every other week. We just want to have a minimum that you can do. If we're all busy, we all have lives. But if you want to do more, and as I said, identify as many invasive species as you can, that's great. You can do that. Also, a question is asked me often. You can, and it's certainly okay to do the rake tosses in the same location every time. You can do one, if you're going to do 3 rake tosses, you can do one in the same location every time and do 2 others in different random locations. It's all good information. If you do it repeatedly in the same location, what that does is starts to lend it to us understanding when things are growing. If you pick an area you've never done before, you don't get any starry stonewort early in the season, and you do later, then maybe it's introduced or just hadn't started growing yet, or maybe a bed is expanding, all those good things. So it's okay to do it in the same location every time or different locations. Starry stonewort is a late bloomer. That's what I call it. Basically, it starts growing. It is growing now, but it's very small. In most places, you're not going to find a lot of it yet, but it grows fast. And later in the summer, August, September, it's at full bloom, I shouldn't say bloom, it's not a flower, but full size. And all of you that have dealt with choked waterways of starry stonewort know that. So there it is. It's not a big deal. We're going to keep it simple for you. So I am going to try right now to, okay, here's pictures of rake toss, people at rakes, and here's a fellow I used to work with us doing rake toss. This is me actually pulling up a big pile of starry stonewort. I'm going to try to run this video. Mike, let me know if it doesn't work right. And this was done by a woman in our office. Who's unfortunately, we lost her to, she went to other employment. She was wonderful, but she did a great job. This was done on one of our local, one of the smaller Finger Lakes.
Today we're at beautiful Honeoye Lake in the Finger Lakes at the New York State boat launch, and we're going to talk about how to do a rake toss. So first you're going to need 2 rakes. You can bind them together side by side with a couple of zip ties. Connect a carabiner to the end, and you connect a rope to that carabiner. And that rope is going to be 100 feet long, and it's going to be in increments a foot between each. You're going to mark off each foot-long section of that rope. So when you do throw the rake into the water, you will be able to judge just how deep it is. When you throw the rake into the water, you want to make sure you secure the end, because you don't want it to go in the water and not be able to retrieve the rake. So you pull it out and you look at all of the plants that you have on the rake and you separate them into piles. Here I'm separating them into piles by species to see what I have present in the lake. And there's some maybe Chinese mystery snails. This is some Elodea, and that specimen there looks very bushy, but then we have another specimen of Elodea. The same plant just looks very different. So plants can look very different depending on where they are in their growth cycle. And then we have this one here, and this one is the same plant as well, only it's on the verge of dying, so it looks very different. We have some Coontail, and that looks very different. That's very brown. And then you'll see there's some Coontail that's very, very green and very fresh. It is much darker. So when looking at plant communities, you have to remember that one plant can look very different. There's a piece of pondweed there. Many different types of pondweed. We've got some eelgrass there up in the left-hand corner, which is a very good source of food for fish. So you always take a white bin with you and you put some water in it. And right now there's some Coontail in the pail. So you take a piece off and you set it in the water. So when an aquatic plant is floating in the water, it will spread apart so you can really look at it if you need to ID it. So a lot of times you'll put a coin next to it for size comparison. So if you want to send a picture to someone to help you ID that plant, they will have an idea of how big those leaflets are. So here's just another look at all of the plants. So I believe we pulled 7 plants from that one rake toss. There's 7 plants that we got out of that sample. And Sometimes there's more, sometimes there's less, but you can see the different ways that plants look. So now we've got another sample in the bin, and that's Eurasian water milfoil, which is an invasive. And you see how I'm spreading it out in the water. I have the coin there for size comparison. And you can see that it has a blunt edge on the tip. It's got a little red tip. The stem is red. Those are some ID factors and when you're trying to ID aquatic plants. So thank you for watching. Anybody can make a rake for a rake toss. Take a walk down to the end of your dock and drop it in and see what you find. If you need help IDing.
Okay, I hope everybody was able to see that well clearly. A couple notes about the video. 100-foot long rope is a good thing, but use what you've got, what you can use. It is a good idea. When I've done it, I've held on one hand, tossed the other hand, and also stood on it because losing a rake is a pain because once you throw out in the lake, you're not going to get it back necessarily. The other thing she talked about segmenting the rope off in a 1-foot section so you see how deep it is. That's very useful, and it's maybe the most correct way to do it. But we are still, after several years, trying to figure out the best way to do that. So don't worry about that too much. You can put little zip ties at every foot and cut them off, but they actually really rough. They tear up your hands when you toss it out. We've tried tape and things, so don't sweat that either. It is useful, because regularly you're going to measure how deep your water is. But that's the basics of the rake toss. Sort out what you found, and then you would report it into EDDMapS, which we'll talk about in a minute. So again, just a quick note on reporting. It's very important. There's no reason, if you go out, it's interesting maybe to find what you can find through your rake toss, but it's vital that the information you found is recorded properly. We're using it because it's a free app for smartphones and tablets. Those of you that don't have a cell phone, smartphone, or don't want to take it out into the field, you can make a note of what you found on paper or whatever and bring it back. And there is a website that Brooke is also going to describe at the end of this. So you can use it from a desktop computer or in the comfort of your home on your laptop or whatever. Again, Brooke Schreier, I may have misspelled your name, Brooke. I apologize there. He's going to work at the end of this and give us a training session about how to do all this. Why are we doing it this way? Well, in the old days, old days isn't just a few years ago, used paper deforms and things and pencil and pen, and they get wet, they get smudged, it's hard to read. Handwriting, and that has to be given to somebody to load into some kind of database. So it's a 2-stage process. It's got lots of potential for error and loss of information and so on. So this is quick and efficient. It's GIS or GPS based, meaning it'll give you a location, so it immediately tags where you are, where you found whatever invasive species it is, and then you can fill in related information about it. So it's quick and easy. Brooke and we can go in and review it for the information you've collected for consistency and potential errors and continuity and things like that. And then we can export it as needed if we need to discuss it in a spreadsheet or so on, or whatever it might need. And then we do update the data, and I got to talk to Brooke about this. We upload the data to the USGS non-indigenous aquatic species database, and we did that because for our grant when we started, we needed something that we put in that worked for this vast region that was several states and part of Canada, so international. And USGS NAS database is a great resource, and they're happy to log in the Canadian data, and they've been doing that. So we'll go there as well. We have a similar program in New York State, of course, and we use a different program here because New York State has its own. It's called IMAP Invasives, and we do that here. But EDDMapS seems like the best route to go for Canada, and that's what we're doing. Great Lakes Basin Survey Day. This is a new thing. There's a picture of the Great Lakes Basin. So we'll have you out there, hopefully surveying as often as you can, or every other week at a minimum. Saturday, August 21st, we're trying to get folks to go out. This is the first time we've done this. Get out there and bring a friend and toss a rake on August 21st and Saturday. And we're trying to do this across New York State with you guys and anywhere else in the Great Lakes Basin. This is based on a sister program that Minnesota and Wisconsin have had for several years. Now, what they do, Minnesota calls it their Starry Trek, which I love. It's a great name for Starry Stonewort. Wisconsin is the AIS Snapshot Day, and several years ago, they decided they picked one day, and what they do is they pick that one day and train people. They set up locations all over each of their states, and they train citizen scientists about how to go out there and do rake tosses and identify aquatic invasive species. We're doing it differently because we've already got you going to work through the summer, doing rake tosses, but we figured, well, let's join on in our own way. So if on top of your normal, if you go out every Wednesday or every other Wednesday, maybe go out that Saturday 21st as well and toss a rake. And so we'll get a lot of people. We're trying to get as many, raise awareness by doing this as a Great Lakes Basin project, spread the word about invasive species, raise awareness about how important it is and how each of our little corner of the Great Lakes Basin is important. And how many rake tosses can we do in a single day? When Minnesota and Wisconsin, they train, I think combined they train over 400 people on that one day every day. And again, it's a very organized thing. It's all over the state. So we'd love to have you do this. We're going to send out more information about that closer to the date. Mike Caston in my office will be sending out, mass emails to describe it and announcements and so on. But just keep that back in your head. I think it'd be really fun if we all get out there, as many people as we can, to toss a rake on August 21st. Okay, questions. I see out of the corner of my eye, I'm looking at another screen here. There's all kinds of conversations going on through the chat box. If there are any questions right now about during the survey, I'll take one or two questions about the survey, about anything we talked to thus far.
There was a question if you could do this in a river.
Yes, yes, you can. In fact, that's a great question. If it's if it's too fast a flowing water body, that's going to get tricky with a rake, and a lot of invasives, including starry stonewort, don't generally, that I'm aware of. This is a question for an expert on this. Go like fast moving water, but if it's a feeder to the lake, or a feeder, if it's contributory to the lake, or an outlet from the lake, absolutely, you can do it there. In fact, we have places down here, work with some local lakes in the Finger Lakes region. Keuka Lake is one, and they've got starry stonewort infestations both in the outlet and in one of their tributaries going in. So it's a problem. So yeah, absolutely you can. Any other quick questions so far?
Nothing that wasn't answered by the video.
Great conversation going on here about Pigeon Lake and so on, and all these locations. I wish someday I can come up and visit you folks and see, because your region sounds beautiful. You're going to get an identification. We've been at this about 45 minutes. I'm going through a lot of information. If you want, we can take a quick breather, take 5 minutes, stretch your legs, and get a cup of coffee or something, and we'll get back into this identification. It's a little more in depth now. We'll be talking about, obviously, how to identify starry stonewort and some of these others. So let's take a quick 5-minute break and come back here, and we'll get going again. Thanks. Okay, folks. Hope they're all back and at least a little bit refreshed. I know I'm going through a lot of information quickly, but we'll do our best here. So again, there's lots of great conversation going on in the chat box. One question was, I know with other invasive land plants, it should not be composted. Is it different with aquatics? I guess I don't actually have solid answer for you, although I haven't heard it's a problem. Most aquatics, when they dry out, they break down into very, very little, there's nothing to them. And in fact, I know that down here, in the past, they've done Eurasian water milfoil harvesting, and they'll take it and they've actually used it in farmers' fields because the stuff breaks down into very fine, once it dries out, there's not much to it, but it's loaded with phosphorus and other things. So I can't tell you, from a professional level, if absolutely it's a problem, but most of the time I don't think it is with aquatics. But that would be something to look into for somebody else. So an expert that would know more about that. I know if you looked at starry stonewort, if you pulled that out and let it dry out, it dries out to almost nothing. It's like this, almost like I don't know what you call it, fragments or dust, this dry stuff. So I think it does compost well, but it's some of it's highly, full of phosphorus and other nutrients. So whatever you do with it, if you dispose of it or compost it, make sure it's not just going to flow back down into your water body, obviously. Okay, so identification. I am right up front. I'm a project manager first. I've learned over the last 3 years or so a fair amount about starry stonewort, but I'm not an expert. Again, Tyler is on today to help us out with this at the end of this section. He's going to point out a few kind of tricky things about comparing starry stonewort to native Chara species and some other things, but I am going to run through some basics about what you should look for in identifying aquatic vegetation, and then I'm going to look at some specific, invasives like as listed here. I'm going to look at those and some lookalikes. And unfortunately, we can't do this live like in the picture. Obviously, COVID aside, it's a long distance, and I couldn't be able to fly up and see all you guys in person somewhere, unfortunately. But so we'll get through as best we can. Again, there's lots of resources available. I put before we came back, I put the document link, which is a link to lots of different documents and resources. Carol said, up on our website, there's all kinds of information as well. So you should be able to get through this. It takes practice. It's like anything. If you've never done this, it takes practice. Don't get discouraged. Just get out there and try it and do it, and you'll be surprised, how fast you start to learn. Learn one or two to start with. Start stonewort and maybe curly-leaf pondweed or whatever it is, and you start to learn more and more. And some people get into it, and it becomes a kind of a hobby, and it's great. Again, the more you identify, the better. We're asking you to do 1, 2, 3 of them, but if you want to get into it and identify lots, that's just fantastic too. So what is a macrophyte? Basically, it's a plant large enough to be seen with the naked eye growing in and around the water. So it's pretty much anything, and that includes starry stonewort, and we'll get into this more too in a minute, but starry stonewort is a macroalgae, whereas something like Eurasian milfoil is a vascular plant. There are differences when it comes to what's clogging your waterway. It doesn't matter so much, but the experts and scientists that are trying to study it and understand how to control it, it's an important thing, an important difference. References. There's lots of books out there for this stuff, and this is a collection that one of our experts in our office, Kate, recommended. Again, you will have this PowerPoint presentation available to you through that tiny URL link. It's up there, so you can scroll down and find this in that PDF file. Although I could, in a follow-up email, I could throw these references back up your way too if you want to try and find these books. The blue book, the Aquatic Wetland Plants, volume 1 and 2 is excellent. It's excellent drawings. Through the Looking Glass, also a very good book. It's highly recommended. The one on the lower left, we highly recommend because we did it. We sent some of these up to you folks through Carol. We sent as many as we could. We've had only so many, and we have to share them with our other areas like New York State. We've got lots of people down here doing the same thing you are. So we sent some of those up, and we also sent a group of those and a group of another bound set of fact sheets that we did that you're all going to have to share, I guess, because like I said, if we get 56 people on, may not be enough for all, but hopefully you can share them within your associations and things. And then the state of Maine came up, or I shouldn't say the state of Maine, it came out of Maine. It's supposed to be a really good guide, and I honestly haven't seen it myself, but it's supposed to be excellent. So how do you identify aquatic plants and things? Well, there's a few basic things, and again, this is not a full-blown identification course by any stretch, but it's some things to learn to look at. Growth habit. Some plants are called emergent, where they come up through the surface. Some are free-floating, floating leaved or free-floating, like your water lilies and things like in the top right picture. Yeah, emergent ones going back, or some things. Somebody actually sent me a picture already. They've got something in front of their dock growing up, and it's breaking through the surface. It's growing up from the bottom and is emerging to the top. You get the floating ones, like the upper right, like your lilies, and then the submerged ones. Starry stonewort is generally submerged. Some places it does break the surface. Most places it does not. So that's one. The growth habit is one way to look at invasives. Identifying characteristics. There's 5 basic things here. Leaves, you look at, and we're going to look at leaves in a minute. There's arrangement, number, and margins between the leaves, and how they're grouped. Leaves are probably the best way, in my mind, for a beginner to, including myself, to identify the invasive species or native species, any aquatic plant. Stems have different shapes between different species. Roots, they have strong roots, they have weak roots. They have rhizomes. Again, starry stonewort has rhizomes, which anchor them to the bottom, but they're not actually a root. They don't take up nutrients like your conventional root would. Flowers, there's different flowers. Obviously, water lilies, we're all used to. They have those great big beautiful flowers in the summer. But a lot of other plants, and I have some pictures of little tiny flowers that if you get into this, you can use those to help identify things as well. And seeds and fruits. That's not, again, that gets a little more advanced in identifying these things, but they are helpful to identify different aquatic plants. So leaves. On the left, there's many different leaf shapes. The top left one, wavy margin, that's actually looks sort of like curly leaf pondweed, which we'll look at. And then there's other ones. You got the margin is that along the edge, if you will, those little different variations on the edges of the margin. So you get wavy and serrated and whole margin where it's not. And then a simple leaf is a single, and then the compound has like little leaflets amongst the single leaf. And then all the shapes. You got threads and linear and elliptic, and it goes on and on and on. There's several different shapes. Starry stonewort doesn't really have leaves. It has these branchlets that are like stalks, and they kind of come in line with that thread shape kind of. Leaf arrangement is really important, and I find that that's actually one of the more useful things to really learn how to understand and identify. You've got alternate, and as you see there in the picture, they're, as I say, they're one side and the other in a slightly lower or higher position than the other side, higher and lower. And those are different from the next one down, where it's opposite, where they're lined up with each other on either side of the stem. Okay, that's different. And then the bottom was whorled, and you may not be able to read that well. W-H-O-R-L is whorl, or whorled, W-H-O-R-L-E-D. And that means around the stem, there's a bunch of leaves that are clustered around the stem in a radial pattern. And starry stonewort, again, doesn't have leaves per se, but it has branchlets, and the branchlets are whorled around the stalk of the starry stonewort. So those are all things to look at when you're learning how to identify these plants. Stems, again, on the left is a round stem, and on the right looks like maybe muskrat or something that's very flat. It looks like a grass. And so there's identifying features, and you look at these things. What is it? Leaf configuration, what do the stems look like, and so on. Roots, we talked about roots. On the left is something with, fairly conventional roots. And on the right is a long, skinny root from a floating, remember we talked about emergent, floating, and submerged. This is a floating, aquatic plant. It goes all the way down to the bottom. Or it floats in the water, collects nutrients. So roots are different. They vary. And remember, starry stonewort has rhizoids, or rhizomes, that are anchored to the bottom but aren't technically roots. And again, flowers. I mentioned this. This gets pretty technical because they're very small in many species outside of the lilies. But again, you can look at the different flowers, the colors, the different petals when they blossom, and so on to help identify, what you're looking at. And then the seeds and the fruits. And again, this gets a little more advanced, but it can be helpful. On the left, it looks like little tubers, and we're going to see that those are usually down in the sediment, like little potatoes that grow in the sediment in the water column from the plant. In the middle, there is that big nasty looking thing. That is the seed pod, or the seed from the water chestnut. And these things are pretty bad. You don't ever want to step on one. They're really sharp, but they can unfortunately survive in the sediment for years and still be viable. But that's another identifier. They have points on them, and they can get stuck in animal fur or waterfowl or other things and be transported to new locations. And then these other things over on the side, we're going to look at in another video I'm going to show in a few minutes. We talked about these in Acara species, I believe. These little growth deals look like little tiny bananas or something. So again, just more information as you go through these and you find something you don't know and you're looking through a field guide or fact sheet. It might be useful to help identify. So we're going to get into some. I'm going to run through a few of these invasives and end, of course, the starry stonewort and go into much more detail about starry stonewort. Curly leaf pondweed. This is a relatively easy one, and it's one of the most easy ones to identify in my mind. It grows up, as we said, emergent. It grows up and then actually floats on the surface. You can get big mats. It can be up to 5 meters long. It has, as it says there, these kind of rigid, long, oblong, reddish leaves, kind of reddish green, and they have this wavy pattern. Okay, it's two wavy margins and a blunt tip, and it's really kind of blunt on the end. It's not pointy. Looks like this. I think it looks out of the water. Looks kind of like some ferns you might see around or something, but it's pretty easy to identify. It does a small reddish green flowers. If you see those, it's already coming up pretty strong now, for sure. And it clogs waterways like all the rest of them. It gets very thick, but it's pretty easy to identify. And really, I don't have a lookalike for it. I don't know if Tyler knows or somebody else. I don't know of any other things that look this much like it that are natives. So again, it's pretty easy to pick out as invasive. Eurasian milfoil. I know you all probably have heard about this. We've been fighting Eurasian milfoil down here in New York State for decades. It looks very much like coontail, which I can look at in a minute, but a giveaway for this is it has a reddish brown stem. And then the leaflets here are kind of blunt. They're cut off. Okay, they're kind of blunt and chopped off. The reddish brown is a giveaway. If you want to get detailed about it, there are whorls. Remember, the whorls are the things that go around the stem. There are usually 4 of them around these leaflets, which have remember I talked about the kind that have single leaf or multi things, like up here, this is a leaflet with multiple little things on it. So there's a leaf with leaflets. There's like 6 of those whirled around the stem. And then, this is a real close up. You can see how brown it is here. And then tiny pink flowers if you can find a specimen now or into late July. So that's a giveaway. It looks very much like, common coontail, which is it's a common native. You find it all over the place. It looks very similar to Eurasian milfoil, but it's all green. Notice there's no red here. Okay, no red. And then these aren't quite as blunt. It comes a little more of a point. So not quite as blunt as Eurasian milfoil. And as the name would imply, it looks kind of like a raccoon's tail in the water. It's again very common. It's out there, and it's sometimes confused with Eurasian milfoil. But in my mind, the way to tell them apart is you go with the stem. Milfoil's got that reddish stem. Hydrilla. Hydrilla is a problem if it gets, hopefully you guys, I've heard that you don't have it much up there yet. I shouldn't say yet, I hope you never do. We get it in New York State now. It's one of the primary things we're looking out for all the time. It grows very fast. It can choke waterways, and it spreads with these little tubers. They kind of grow laterally into the sediment and then pop up somewhere and grow. The leaves and stuff and stalks will grow to the surface, up towards the surface. The way to tell them apart, though, is you got to look really closely, but they have these serrated edges. I hope you can see this in the upper right corner. I'm going to show you a picture of Elodea in a minute, which is a common native. They're smooth, but these have the serrated edge, okay? They usually have 3 to 8, but usually 5 whorls. Again, that whorl, that's important. So if you look at 5 whorls, you get the serrated edge. And if you do get into the sediment, these little tubers, it's possibly hydrilla. And down southern United States, they have a couple different varieties of hydrilla, and it's a real problem. This stuff grows very fast. You can choke a waterway. So Canada waterweed, also Elodea, and I believe there's other species of Elodea out there. It looks very similar, but they're smooth leaves. They only have 3 instead of 5 or more. They only have 3 leaves and whorls around the stem. They do have tiny little flowers, if you can see them. It looks, again, it looks like hydrilla or Brazilian waterweed. So again, I'll back up again. These are smooth, okay, smooth on the edges, maybe 3 around. And then this one is got the jagged, serrated edge, and there's more of them. There's 3 to 8, usually 5 around, whorls around. Hopefully you don't have hydrilla up here yet, up where you are. Water chestnut. This one is, if you've ever seen a pond or lake that's choked with this stuff, it's kind of unbelievable to see. It grows on the surface. There's hardly anything you can confuse it with. There's very few lookalike natives. Again, it's got these really sharp seeds up here. It grows in these florets. You get these jagged leaves. They're kind of glossy, triangular shaped. It's got the stem or petiole here is very thick, flated kind of. They have little tiny white flowers at certain times up here, but right here, somebody's holding one. They've got one of these florets in each hand, right? So they grow in these like little bouquets of invasive species. They're easy to see. They float. The one good thing about them, though, is we have out of our office, we've managed some programs, or my associates have managed programs where we've actually eradicated this from some ponds and bays and things. And it's taken basically a lot of organization, a whole bunch of volunteers and waders and kayaks towing little boats, and we pile the stuff in and take it into shore. And they just, at the right time of year, you pull it up before the seeds form. And we've done that over several seasons in these locations. It can take 2 or 3 seasons, but we've eradicated it in some areas. So even though it can completely choke out a pond, they can be eradicated fairly easily, but it takes a lot of work. I mean, it's easy, but it's a lot of work and just physical labor pulling the stuff out. And they pull tons of this stuff out of there. But again, it's good to know that, when you have a win is a win, and we'll take it, but this is what this stuff looks like. Whoop. Okay, our favorite, starry stonewort. Here's some pictures. I'm going to go through this fairly quickly. On the left is an often used picture by Paul Skowinski. He's a researcher out of Wisconsin. You get how there's a nickel there for size. It's a very stocky looking plant. Again, I misspoke. It's not a plant. It's a macroalgae. It came from Europe and Asia. It was first identified in the late 1970s in St. Lawrence River, probably from the ballast of ships, like many invasives have come in this region. And it's spread throughout the Great Lakes Basin, and it's long and stocky. Doesn't really have leaves, but again, here's like right here in the middle. If you can see this, there's this whorls of 4 to 6 around the middle. Okay, so and these are my dirty fingernails. I was out in the field first year here, and these are the star-shaped bulbils, they're called. Okay, they're not really seeds, but they're reproductive structures that are usually down in the sediment, and they're star-shaped. And generally speaking, once you see the star, then you've got starry stonewort. However, Tyler's been doing some interesting research with his doctoral work, and he has some potential exceptions to that, and he may clear that up for us a little bit. Generally speaking, though, the star is a giveaway that starts stonewort. And this is just an underwater shot of a really big bed of it given to us by Scott Brown from Michigan. Very helpful. Impacts. Impacts are very much, you know what the impacts are. I probably don't have to tell you, very much like many invasives, rapid aggressive growth, environmental systems, they mess up biodiversity and everything. Interestingly, most fish, and I think research has only found 1 or 2 species that will even spend time in this stuff for any length of time. It's too thick in things for fish to spawn in. They don't even use it for cover much. I've heard from fishermen that Eurasian Milfoil does provide decent cover for fish, but starry stonewort does not. It's really bad for, it doesn't, pushes them out so they don't spawn in it, they don't hide in it, and so on. The question of what it does with water quality is still up in the air, although there is some evidence that it actually might help water quality. And then it's a possible phosphorus sink. In other words, it takes up phosphorus, but there's research ongoing with that. And like many invasives, it just kind of replaces native species. This is a shot I took in the outlet of the lake I told you about. It's solid starry stonewort. There's more over here. And there's the lilies that are hanging on still, but it's a big solid mess of it. Human impacts, again, it messes up the use of the waterway. It's hard to pull your boat through there or even kayak through it if it's really thick. You don't want to swim in it, certainly. Fishing, as I said, it's hard to pull a fishing lure through there, and there's likely very few fish anyway. So then you have your potential economic impacts from all those things. So it's just as bad as many of the other invasives. What do we know? Again, I think I mentioned this, starry stonewort, there's a lot we don't know about starry stonewort compared to other invasives. And there is a tremendous amount of research being done on learning more about the life cycles, what are the limiting factors in the environment, even if you're doing algicide treatments, what algicides work best on it, and so on and so forth. And why does it grow in some places quicker and not others? There's just a million different things. There's a lot we do not know about starry stonewort yet. But the good news is there's lots of research happening to try and figure this stuff out. Current extent, this is a little bit old, I apologize. These little red dots are from over a year and a half ago, I'm afraid, I apologize. Your area right here, we know there's way more than is showing here. So for example, there should be a lot more dots through here. But you know, it's largely confined. This was it, you know, it's from the USGS database. So far it's still in the Great Lakes Basin and just outside, all over as far as Vermont, unfortunately at this point. So hopefully it won't spread too much further in the US, but you know, Michigan's got its hands full, and New York State, we've got our hands full, and you guys certainly do. And again, this is all that's been reported. So we know we're learning quickly over last year and this year working with you guys. You're finding more and more areas that just have never been reported, and there's just a lot of it through here that isn't showing in this database at least yet. So I'm aware that it's here, but you get the idea. It's spread quite a distance since the 70s. So some more pictures. Here it is, kind of laid out, a couple individual strands on top of a kayak. So it gets quite long. Here's again the whirl where the branchlets are looped around. Here's, if you see this up in the right-hand corner, that's a little string of bulbils on the rhizomes, which rhizomes look like fishing line. I think Carol said she looks like rice noodles, which is a really great description. So that's, you know, dig this up mostly from the sediment. They can grow up to 2 meters long, 4 to 6 blunt, you know, branchlets around each. Again, around in the whorl, around the branch, around the stalk. And it is similar to other stoneworts, which are native and okay, but you know, there's a tiny star-shaped bulbil, as we've been talking about. More pictures. This is kind of, you know, this is in like a kind of a dish in a lab or something, but you can see, you know, the stalks and the branchlets. These are some of the technical names, the nodes, the branchlets. Internodal segment goes between the nodes. The bract is kind of an extra growth that comes off. It's like a little mini branchlet. And the video I'm going to show in a minute, he mentions bracts. My understanding is, and I'm not the expert, but these macroalgae have some of the biggest plant cells in the world, in nature. And, you know, the cells span these long distances, long skinny cells. They take in nutrients and things through the stalks and the branches and things, which is why they're different from a vascular plant. It takes in sunlight and things to the leaves, but takes in most of its nutrients to the roots. And then here's another picture. It's a little fuzzy, but you see how long and stocky this thing is. Lopsis obtusa, should you call it that? That's the scientific name. There's more pictures. These are from a harvester in Keuka Lake near us, and you know, I can guarantee you that most of this is starry stonewort. I was pulling these rakes up, and these are 90% starry stonewort in this bed here. I showed you this picture already. So again, not good pictures. I know you've got your own horror stories and pictures from where you are. This picture came from Carol. Again, I'm picking on Carol today, but this is a beautiful picture of just a big glob of starry stonewort they pulled out. But this is, you can see the long, thin, clear rhizoid. Looks like, as she says, rice noodles or fishing line. And then you can see there's bulbils, you know, scattered or not scattered or lining here. They're in a row. So I'm trying to say a row of them along the rhizoid. This is a picture Carol sent me of her poor little lake choked with the stuff, and this is an underwater picture of from again, our friends in Michigan that just shows it gets very dense. It gets super dense and thick, and is a problem. So it can be confused with Cara, and again, I'm going to show a quick video, and then Tyler's going to go through more of this. Muskgrass, you can be sometimes confused with this, but you can see the difference. It's not, Cara and muskgrass are not quite as long and thin and stalk-like. It's got the whorled branches and things. One big thing I've done to learn to tell what it is, because sometimes you take it out of the water and it doesn't look quite like this. If you pop it, now this is also a macroalgae, so again, it's built different. Those long, skinny internodal segments have cells. You can squeeze it and they actually break or pop. And if it smells like garlic or musty or skunk-like almost, it's going to be Cara or muskgrass probably. Starry stonewort does not stink when you pop it. So I've used that and it's very helpful. It's a real helpful little trick to tell the difference between them. Sago pondweed is another native that's a good plant to have around, and it can be confused with starry as well, although it's, you can see it's a little more, it's got like the kind of feather-like or fan-like, and it's all these long, skinny, remember we looked at the leaf structures and those thin needle-like leaves, these long, thin leaves, a little denser there. Kind of look like it's a fan or a feather or something, so it's a little bit different. So I am gonna hopefully show a video by, again, Paul Skowinski. He's with University of Wisconsin Extension Lakes Division up in Stevens Point. He did this video. He's an expert in all kinds, pretty much all northeastern aquatic plants. Great guy. He's been very helpful, supportive of the program. And this is like a little 4-minute video. Note as he goes through this starry stonewort and the native lookalikes, he has smiley faces and frowny faces for the natives good and the bad starry. But it's a nice little video.
Starry stonewort is a species of macroalgae related to many native species in Wisconsin. This species can be challenging to identify, but learning a few important features will help you distinguish starry stonewort from its relatives. These algae have a simple body plan consisting of a main axis, which you could consider the stem. For simplicity's sake, we will just use the word stem in this video. Around the stem are whorls of branchlets, much like the whorls of leaves you would see on other aquatic plants. A key feature of starry stonewort, and probably the easiest to look for, is the presence of star-shaped bulbils. These are produced on clear threads in the sediment, which look like fishing line. A small number of native species will produce bulbils too, but these are generally round like tiny snowballs. Starry stonewort bulbils have many points or lobes. Starry stonewort is very large compared to most other macroalgae in our region. Notice the size of these native species compared to the nickel in each photograph. Then look at the starry stonewort. It is several times larger in diameter than most of our native species. Starry stonewort produces small orange balls along its branchlets. These are the male reproductive structures called antheridia. So far, only male starry stonewort has been observed in North America. This means that there is no sexual reproduction occurring, and therefore no viable seeds are being produced. Without females present, starry stonewort can only spread by bulbils and fragments of itself. Starry stonewort is likely to be confused with several native species. The most commonly confused species are the muskgrasses, also called chara. However, all of the common chara species have long cells running up and down the stem, creating a rough or textured feel. The stem of starry stonewort is smooth. Species of Nitella can also look very similar to starry stonewort. The branchlets of Nitella always fork at the ends, producing 2 or more equal length parts. The branchlets of starry stonewort may appear to fork, but what you see is actually a long bract coming out of the branchlet. Since these bracts are often only on the upper branchlets, you can look further down the stem where the branchlets don't have any forks or bracts. When starry stonewort is removed from the water, its stiff branchlets tend to remain in the same position. Species of Nitella tend to be very relaxed and droopy when they are out of water. Sago pondweed is a common native species that can look somewhat like starry stonewort. However, notice that its leaves alternate left and right on the stem rather than being in whorls. It also produces many large seeds on long stalks above the plant, which starry stonewort would never do. Horned pondweed has opposite or whorled leaves, but will never produce bulbils, and the leaves are very weak and delicate. It produces many clusters of 4 banana-shaped seeds along the stem, which it typically holds throughout most of the growing season. Remember, without females present, starry stonewort can only spread by bulbils and fragments of itself. Boat trailers, anchors, and other recreational equipment are the most likely vectors for spreading starry stonewort. Monitoring boat landings is a smart strategy for detecting an introduction of starry stonewort and slowing its spread. For help identifying starry stonewort and other aquatic plants of your lake, check out the field guides and other helpful resources.
Okay. All right. So that's that video. It's excellent. You in those resources, and we'll send it to you again. You can look at this yourself anytime you want, but it's up on YouTube. You can watch the video again. It's very helpful. So now, Tyler, you're still with us, I hope. I want to invite Tyler to he's going to give, I'm going to stop sharing my screen, and Tyler's going to share his, and he's just got just a few slides. He's going to talk a little more about comparing starry stonewort to chara species and some other things that he's found in his research. Again, he's with Dr. Andrea Kirkwood at the Ontario Tech University. He's working on his doctorate up there. He's done all kinds of monitoring and sampling for starry stonewort. So I'm going to stop my share. Whenever you're ready, Tyler.
Yeah, just trying to. Can you see that okay?
Looks good to me.
So, hi everyone. Like David said, my name is Tyler. I'm a student in Dr. Andrea Kirkwood's lab at Ontario Tech University. So I'll just start by sharing this. So in May, this marked my 6th year studying starry stonewort across Ontario. And what I hope to impart on you today is a lot of the mistakes I've made and learning progress that I've actually made for starry stonewort in Ontario. Because what we largely found was much of our headaches, starry stonewort is not behaving like it was in the United States. So we had to constantly adapt and change our sampling as well as laboratory processes. This is largely due to the heterogeneous landscape of Ontario. So as you can see here, all the complex colors as you go north into the Kawartha Lakes, this is a shift in the underlying current geology, which ultimately influences the water chemistry and the taxonomic or appearance of starry stonewort in these lakes. And one of the biggest things that we found was in the United States, starry stonewort is what you almost can say is a dose-dependent response on calcium. The more calcium you had, you generally saw more starry stonewort. But going across on central Ontario lakes and actually modeling the environmental parameters to starry stonewort presence, we found that calcium was the lowest predictor, which again proved to be a huge headache and often very confusing. Luckily, we had research collaborators from France and part of the native range where starry stonewort is, as well as Joe Lattimore from Michigan. She was very, very helpful in correcting a lot of our early mistakes and stuff like that. So one of the largest things that can be a problem for starry stonewort in Ontario is in the region that you guys currently inhabit, stretching from Georgian Bay to, I'll say Kingston, there's about 25 to 30 or so different native Macrophyte species that are often very confusing to differentiate between these and starry stonewort. And for various reasons, now in 2021, or starting in 2019, we're starting to see a huge increase in these populations, which again, can confound some of our results, prove very confusing and stuff like that. So I have 3 photos here of Macrophytes from Ontario, and I think if Shelby's here, I'll pick on her just to try and highlight the difficulty in differentiating between some of these species. Because at a quick glance, sometimes these 3 images here would be classified as starry stonewort, and they may or may not be true. Within the United States, as well as Canada, and even in the native range, identifying starry stonewort correctly is very difficult based on appearance. For instance, there's a case in Japan where starry stonewort was classified as extinct. They thought it was gone. And then when they started doing DNA analyses of Macrophytes to try and combat some of the population changes they were seeing, they actually realized that starry stonewort was still present. It just changed its appearance. So are you here, Shelby, or?
Yeah, I'm here.
Okay.
Let's go.
Yeah, do you want to take a stab at these? And
Sure. So I don't believe A is starry stonewort.
no, you know what it is?
I think it's Tolipella.
Yeah. So like David said, Tolipella is very common across Ontario for the region that you're going to be sampling. And this is often confused with starry stonewort. You want to try B?
Yeah, so B is also not starry stonewort. I think it is Cara.
Yep, another one that David pointed out. And then you want to try C?
Yeah, and I think the last one is starry stonewort. No?
No, so this is actually Nutella, which is another species that David pointed out is commonly misidentified as starry stonewort. It's close. It's really difficult sometimes to tell, especially from photos. And we saw this in our work where even when we threw a rake and took a sample, sure, there might be a star present, but when you actually take it back to the lab, wash it, and actually start sorting through it, you realize that the huge mass you thought was starry stonewort is actually a very small fraction of that. I have a few more photos here. So like the video pointed out and David was talking about, Macrophytes have these bulbils, which you can see in the left image here. Pretty much every Macrophyte can produce bulbils. Normally, like the video mentioned, that they can be circular, but because Ontario is so heterogeneous and has such a diverse native Macrophyte community, you actually have instances where the bulbils, like this image here, can sort of look like the star. And based on the environmental conditions, you can see small changes in the appearance of these, not as large as you can for the stem and the branching morphology, but it's still important to note. So this bulbil here, just because of the little outcrops and stuff like that, could be mistaken for starry stonewort, but it's not actually starry stonewort. And another thing to point out is this is common across like all Macrophytes. So for instance, when we have samples like this, even sometimes I don't know, and I need to get more information, even though I've been doing this for 6 years now. And what we found was conservation authorities all the way up to federal agencies, when we bring them into the lab and actually start pointing out these differences, it becomes evident that the misidentification seen within Canada, the United States, and even the native range is a big problem. So I know David's mentioned this before, but we also have an email that we set up. So if you ever encounter any situations where you might be confused, please don't hesitate to ask. We're always here to help. This is just a photo that I wanted to highlight. So this is a Tolipella species similar to the one on the panel A here. But Tolipella in the later growing season, it's almost like they have these little bulbs like you can see right here. So initially, earlier in the season, because charophytes are generally what you call thermal tolerant or boreal taxon, some charophytes, including starry stonewort, can actually live under the ice. So we've actually seen it persist as early as March in huge populations comprising more than 90% of the macrophyte community. But another thing, so you're sampling from July to October, where you can rely on these to maybe differentiate between any Tolipella species that you encounter. So later in the growing season, usually around late July, early August, Tolipella will start producing these, I call them almost like rosebuds, very dense and entangled thalli. And you can clearly see that they're much different compared to the main thalli axis right here or even down here. That would traditionally be confusing for differentiating between starry stonewort and Tolipella.
I have a question. What's the common name for Tolipella?
Bird nest stonewort.
Okay.
But it also depends on the species. So for instance, there's also fragile stonewort, which is Tolipella intricata. It really depends on what you're dealing with. Another photo that I just wanted to highlight really quick. So this is from Carol Cole. I thought this was really interesting because she sent this, it was a week ago. I sort of lost track of time recently. But like I mentioned, because mainly three factors, since 2019, we've seen explosive increases in the charophyte communities across Ontario. But starry stonewort generally has a characteristic disheveled or unkempt teenager looking vibe going on. So if you ever encounter stuff like this, an easy way to tell if something's starry stonewort is, if you can see my mouse here, this clump right here seems more like disheveled, not as nice looking as the native charophytes over here might be. So that's also something to keep in mind. I think it's mostly from Michigan, and I believe one paper in Wisconsin generally says they see similar things where starry stonewort is disheveled and it has sort of like, it's not as neat looking as some of the native charophytes. I just wanted to draw your attention to this figure here. So because of Ontario's unique landscape, unfortunately, so like David pointed out and the video pointed out, usually Nyctalopsis or starry stonewort can have very large cells. They can grow upwards of 2 meters. But so if we just look at panel B and C here, these are actually both starry stonewort samples taken from Lake Simcoe within 100 meters of one another. So you can see just based on this, how the environmental parameters influence the appearance. But again, sticking to the identification guide that Kawartha Conservation made for you guys, the video and all the resources that David's provided, you can start to understand the differences between these. But if you're still not sure, like I mentioned, we recently created this email address here. And here's another instance where starry stonewort, this one here, might be misidentified as a Cara species, but these two samples are actually starry stonewort from Lake Scugog. So like I was mentioning, if you still are unsure or just want to get more information in general, don't hesitate to ask us. You can just send us an email to this address here, and either Shelby or myself or Dr. Kirkwood would be happy to help you guys further.
Wow.
Was there any questions?
Thank you, Tyler. That was fantastic. I will say, if anybody's thinking about questions, so it sounds a little daunting, but it's important, and don't give up. I mean, try, as Tyler said, get out there and do your best to identify these with the resources you have at hand. We have lots of resources for you. And Tyler, if you wouldn't mind, could you put the email address in the chat box?
Yeah.
That'd be really handy. So yeah, don't despair, folks. Get out there and identify stuff anyway. Do your best. It's very important. But it's great. We've got Tyler, and Dr. Kirkwood, and others that are really looking at stuff in detail. Because in the big picture, this will help us get a better understanding about starry stormwort, how to control it down the road, and those kinds of things. Any other questions for Tyler? Okay, I'm going to share my screen back again. So this is kind of my last slide before we go into actual training in how to use EDDMapS. Again, I put this, speaking of resources, I put this in the chat box. That's, it's kind of a link to our all those files I showed at the beginning that we set up for this training. Number one here, links, references, and contacts. You can download that and email it to somebody if you want it, and they'll put them back in that location again. It has a PDF version of this actual presentation, links to the videos I showed you, and then some links to various field guides and fact sheets and other information we have on our own servers at the Finger Lakes Prism. And then our website, of course, starrystormwort.org. This has got the library there and a bunch of other information. And we have 40 some odd documents, presentations, and related. In the library, everything from quick presentations and fact sheets to we just got several full-on papers from Wisconsin, I think, or Minnesota. Thank you, I forgot which state it was, but anyway, we got lots of information up there. Again, any questions before we get into EDDMapS training with Brooke? Douglas has asked about is an app that identifies plants and trees. I don't know. I'm not sure what you mean. I'm going to guess that it doesn't have a lot of detail on aquatic plants. You can try it, but I wouldn't necessarily depend on it.
I would maybe just add for the app portion. What we've noticed recently with iNaturalist is a lot of reports of starry stormwort for Ontario are actually not correct. So again, what I would recommend is if you're still unsure about something, take as many photos as you can. Send them to David, myself, anybody, any one of the emails that you've received today, and we'd be happy to take a look at those.
Yes, and I would say also, just from the point of view of our staff reviewing what you've done, and Brooke, we'd almost insist on taking photos in the EDDMapS. You take photos. It's kind of we're going to insist because if there's no photos, we can't confirm it, and then we're kind of lost. So take photos. Don't download photos. Take your own photos of what you found.
Answer this question as well. So about the residents clearing these invasive weeds, I would not recommend removing starry stormwort from your property or anywhere else. Just based on the Macrophyte life cycle, and because they're so abundant, when you actually remove plant material or any competing species that might be caught within the network, this actually primes the ecosystem to allow new populations to develop from the calcified garogonites and sediments. So what I would recommend doing right now is just try and do your due diligence, make sure that the identification is correct. And I believe Brett Trigano from Fourth Conservation is doing a pilot study for these bubblers that might prove beneficial for reducing weeds on properties. So if you want more information about that, I would reach out to Brett, and hopefully this can help you in the future. But again, I wouldn't recommend trying to pull or clear any of the weeds at this point in time.
Yes, I agree. They fragment easily, and we don't want to start new or expand existing stations. Okay, with that, we're moving on here to time. I don't want to go forever. Brooke, if you're on still, we can take some more questions at the very, very end. I'm going to save this. I believe we can, Mike, if you're still on, we can save the chat window to a file, and then we can try and address some questions that we didn't get to with the email and so on. So Brooke, if you're there, I'll stop sharing and you can start sharing.
Awesome. Thank you.
There's Brooke.
So let me know when you've given me access there, David.
Oh, didn't I give you access? That's embarrassing, right?
It's all right.
Sorry about that.
Happy Wednesday, everybody.
Yeah.
Mhm.
Should be good.
Awesome. Thank you.
All right, thanks, Brooke.
No problem. All right. Hi, everybody. My name is Brooke Schreier. I work with the Invading Species Awareness Program out of the Ontario Federation of Anglers and Hunters. So, David, thank you, by the way, David, for including me. And, I've worked quite a bit in conversation with David over the last year, trying to situate ourselves within the Starry Stonewort conversation. And I also have to say thanks to Carol Cole for always facilitating that and connecting a lot of the dots, connecting me with Andrea. And I'll kind of go into, today what I'm going to do is, for the sake of time, I'm going to go fairly quickly through EDDMapS and how you guys should be reporting your Starry Stonewort observations or your suspected Starry Stonewort observations. And I'm also just going to kind of give you some higher level stuff around other reporting tools that you can use in addition to that email that Tyler gave you guys. So right off the hop here, on the left there, you've got Chinese mystery snail, which is an invasive snail to Southern Ontario. Was introduced in the late 1800s and is something that you might encounter while you're out on the water. So, take that opportunity as David really alluded to. And if you see things that you suspect are invasive species, capture some photos and you can report them via EDDMapS, just like you can the Starry Stonewort. David already touched on a lot of this, so I won't really go into it too deeply, but the importance of citizen science and why the things that you guys are doing is so important in terms of invasive species. Then I'm going to go into reporting tools in Ontario and then how to report invasive species, which is really the meat and potatoes of the presentation today. David already had this. So just to reiterate, citizen science is so important because it helps us in those early stages of the invasion curve, right? Really trying to prevent introductions through education and outreach, teaching people what to look for, just in case something is introduced. David highlighted hydrilla. That's a species which we don't have here in Southern Ontario. And obviously there's a lookalike species, Lodia canadensis, but at the same time, like, let's say one of you guys happen to actually find it, then we've hopefully detected it early enough in this invasion curve that we can then subsequently eradicate it. So it's really important, and the work that you guys are doing is very important. So the invasive species hotline, right off the hop, I want to start off by saying, I know some of you may not be very techie, you might not feel comfortable with downloading an app or using a smartphone. And I'm also going to walk you through reporting on the web page. You might not even feel comfortable doing that. So worst comes to worst, what you can do is you can give us a call Monday to Friday, 9 to 5, 1-800-563-7711, and say, hey, I'm part of the surveillance program for Starry Stonewort. I took some photos, maybe just on a camera, and I want to get some photos into you guys. At that point, pretty much the bare minimum of technology that you're going to be asked to use is email. You're going to be asked to email photos to us, at which point you can just give us the sighting location and then we can upload it to EDDMapS for you on your behalf. So, like kind of bare bones technology, this is the best that we can do in terms of, getting those reports in. So make sure you give us a call if you have, some questions or if you want to submit reports this way. I'll kind of spare you with all the details around the hotline, but let's just say it's very busy. So you're probably going to have to leave a voicemail, and we will be sure to call you back. Jumping into EDDMapS. So what is it? It's an application for both iPhone and Android. And as David said, you can actually download it on, things like iPads. So if you're more into using tablets, that's fine as well. We track almost 200 species in Ontario, which really only represents 30 to 40% of invasive species that we currently have in the province. There's over 445 terrestrial invasive species in Ontario, and anywhere between 30 to 40 aquatic invasive species. So there's lots of them here, but for the purposes of tracking in Ontario, we really keep it down to around 200. To date in the system, we have over 54,000 confirmed reports, and that's since having this platform adapted for Ontario. And every single year, us in-house, myself, my colleagues, we have a number of protocols, which I'll walk you through real quick. And we go through, thousands of confirmed, vetted, and released reports. So every single report that gets sent in gets reviewed, and questions get asked. There's back and forth between reporters and us to ensure that any data that's going into the system is accurate. And in terms of that accuracy, with starry stonewort, as you just saw, and by the way, Shelby, I also thought the third one was starry stonewort. So you're not the only one that got it wrong. It can be a real challenge, right? And that's what we're experiencing here today. But that being said, I'm happy to say that in speaking with Carol and the University of Georgia, who actually runs EDDMapS, we've added Dr. Andrea Kirkwood, as well as Tyler and Shelby as identifiers, as verifiers on EDDMapS. This is the first time this has happened since we had it adapted for Ontario. So what that means is, when you guys submit your reports via EDDMapS, Tyler will be the one looking at it, right? So he'll be the one that ensures that it is the correct species. Because personally, I would just be a middleman at that point. I would be sending those photos to Tyler and Andrea for the expert identification. So we've just made it easy so that, they're just able to identify it for you guys. That being said, at the end of the season, once all the reports are in and they're all shown on the map, you guys can download that data. So you can actually go to eddmaps.org and download all the starry stonewort data for Ontario if you're curious, or even just look it up on the distribution maps, which are on the website. And I'll show you some of that in a little bit here. Also want to speak real briefly about iNaturalist. iNaturalist is great, but for the, and we have a project in iNaturalist. We have over 84,000 observations of invasive species in the province alone, and 525 members in this group. And it is a great project. However, one of the limitations, especially when it comes to starry stonewort, that we noticed was that because, it's a community identification tool. So what that means is if you were to upload a photo, other people on this call could be like, yep, that's starry stonewort. And then it'll become research grade, right? And it doesn't really necessarily matter if an actual expert has looked at it, which is a good thing and a bad thing in terms of the tool. So that's why, with this particular program and the intricacies that go along with identifying starry stonewort, we said, let's use EDDMapS, because then it has to go through the verification process before it gets released as starry stonewort. So still a great program. Still encourage you if, in your downtime, you still want to upload your sightings, maybe after you've received confirmation that it is starry stonewort, then maybe upload them to iNaturalist. So we have reporting protocols within the program, right? So this is what we do. We are actually a partnership program between the Ontario Federation of Anglers and Hunters and the Ministry of Natural Resources and Forestry. We've been doing it since 1992. So we're the front line on invasive species in the province. So when people call or email or send in EDDMapS reports, we have a number of protocols in place. Everything from like pre-screen protocol for handling things like, goutweed, which are very low priority species. We have, Asian carps. So reports of Asian carps in Ontario's Great Lakes. Priority aquatics not known in Ontario, like northern snakehead, Channa argus, which is actually found on the eastern coast of North America. You've got other AIS reported within their, or outside of their known range, like round goby. Then you've got other species like mile a minute that doesn't exist. Asian longhorn beetle, if you guys have ever heard of it. So essentially, we within the program have protocols set up for all these different species in the province. So dog strangling vine and wild pigs more recently. So going back to, that citizen science piece, though it's not necessarily the case with starry stonewort, given that it's been in the St. Lawrence since 1978, it's kind of well established throughout Southern Ontario, there's still an opportunity here to facilitate early detection in water bodies that doesn't otherwise have starry stonewort that we know of, right? And to kind of reiterate the importance of citizen science, because this is what the protocol looks like. I'll spare you guys the details here, but there's flowcharts for all those different protocols and how we handle it and how we follow up. So with Asian carps, for example, this is what citizen science leads to, right? There's people on the water who are aware of these invasive species who report them to us. And in circumstances like this with the grass carp, which is an invasive, Asian carp, we were actually able to pass that information along to Department of Fisheries and Oceans. And they're actually able to get out there within 2 days and get rid of 10 grass carp in our water. So, connecting all the dots, there's a lot that goes on behind the scenes. Like when you submit that report to EDDMapS, there's still a lot that goes on behind the scenes. So, it's great work that you guys are doing. So let's jump into the biggest part of why I'm here. And I'm here to train you guys on EDDMapS. So we're going to start with the application. So I'm going to walk you through kind of really detailed overview of how to download, how to report. And then after that, I'm going to jump out of my presentation, go to a web browser, show you online how to do that. And then at which point, if there's any questions, we can do that. Now, before we jump into this, again, like this is just one other tool for you guys to use in reporting the starry stonewort or other invasive species. You still have, starry stonewort ID@gmail.com, which will go directly to Tyler. Even if you want to email me or if you want to call the hotline, 1-800-563-7711, there's, different levels of comfort for all the users who are going to be doing this. Okay. So first things first, you've got your handy dandy smartphone, your iPhone or Android if you have one. And what you're going to do is you're going to go to your App Store or Play Store or whatever the heck it's called, and you're going to look up EDDMapS. Just look up EDDMapS. I had a question earlier in the presentation about EDDMapS Ontario, and real quickly, EDDMapS Ontario is actually being phased out. We are actually being amalgamated into the bigger EDDMapS platform. That's both the app and the web browser. So right now, when you go to submit a report, which I'll show you on the web browser, it will transfer you to EDDMapS Ontario, but I'm waiting until essentially the end of this summer before EDDMapS Ontario ceases to exist. So I figure for you guys, it's just best to be ahead of that curve and be focusing on just EDDMapS. Don't use EDDMapS Ontario. It's just keep it simple that way so that you don't have to do that change when the time comes, okay? So you're going to search EDDMapS in your store. You're going to see this, this thing right here. You will probably see EDDMapS Ontario, avoid it. Charles T. Bargeron, that's Chuck. He's the one who developed EDDMapS. So, you'll know that that's the right one right there. And then you're going to click on the download button right there. It'll look a little bit different for you if you've never downloaded it. Click on it and make sure you're on Wi-Fi. Okay. Make sure that you're on internet so that you're not using a bunch of data to download the app. I did that once and I actually went over on my cell bill. So try to avoid that. Then it's going to show up on your home screen once it's finished downloading. So it's right there. That's what the icon looks like. You're going to click on it and it's going to bring you to the left page here. And it's going to ask you to use your location. And I know how a lot of us are paranoid around our phones and everything tracking us. So, or at least that's maybe just me. So what you're going to want to do here is actually just click on laser pointer, allow while using app, right? So when you're in the app, it's tracking where you are. Okay. So you're going to click on that. And that's important for submitting reports. And I'll come back to that a little bit later. Then you're going to get this pop up. So this is all sequential, by the way. This is exactly what you're going to go through when you download it on your phone or on your tablet. It's going to say, we'd like to send you notifications. Totally up to you whether you want that or not. It'll send you reminders. I personally don't like push notifications. I find that they always go off at the most inopportune time, like when I'm in a meeting or something. So I usually just, silence all apps. So then you're going to get these, nice little dialog boxes, which are kind of nothing too crucial. It's just going to go through them one at a time. Tell you all this cool stuff about, how important it is to take a good photo. And I think that's been reiterated a few times here today by Tyler and David. And I'm going to say it again, take good photos. It's going to record your GPS location, right? And that's again, why it's so important that you allow it to track you while you're using the app. Looking for real-time, we're doing real-time tracking here, right? So as you submit reports, it's going to come to us. And if we release it right away, it's going to be in the system within minutes. And then, as I said, there is a verification process. Tyler will be helping with this. Dr. Kirkwood, I think Shelby will be helping. I probably won't be. I'll just leave it to those guys. If they go unreviewed, I might send them a reminder, say, hey guys, there's some Starry Stonewort reports that you should review. But each one is going to be looked at. So then next, it's really important because this app that you're going to download is for all of North America. It's going to be really important that you select the proper jurisdiction, right? That goes without saying. Make sure you select Ontario. And then you're going to come to the homepage. And this is what your main page is going to look like on your EDDMapS app. Okay, you've got new sighting, negative survey, upload queue, and then your different taxonomic groups. And we'll go over these in a little bit. Let's first jump into the settings, okay? Top left, click on that. It's going to take you to the settings. And this is really important because you have to log in, right? Now, if some of you have already logged in or created an account with EDDMapS Ontario, that is okay. Those credentials will still log you in here because, again, it's all the same system. So you're going to go to the settings. You're going to click on EDDMapS Login. And then it's going to take you to this page here where you're going to see email, password, or log in. And if you are a returning user, just plug in that information, those credentials, and you're good to go. Now, if you're a new user, you're going to click New to EDDMapS, and it's going to open up this page. The only thing it's going to ask you for, the only things that you have to submit are your name, an email, and a password. Okay, everything else is optional. That's just so when you submit reports, we can follow up with you via email if we have additional questions. Like let's say you submit a report with no photos, we can email you and say, hey, we require photos. Can you email us some photos for this report? So that's how you're going to make your account. And then it's going to take you back to this page here, your settings page, and just go into settings real quick. Make sure the app is working the way that you want it to. So it's going to bring up this page, save photos to camera roll. I don't do it because then you just get a ton of photos that are filling up your storage on your phone. You can auto upload for reports. Also, do not do that because what that means is while you guys are out on the water, when you make a report, auto upload means that it's going to auto upload it to us, which means it's using your data. Okay. You don't want it to use your data. You want it to go into a queue, which it'll do automatically if you don't have that selected. So keep this unselected because then I'll show you at the end of the day how you submit those reports. You can also set up weekly reminders. So just in case, you have 10 reports in your queue, it'll say, hey, you've got reports in your queue. Do you want to upload them? And then obviously it's got your location and that's about the most important stuff there. Okay, so now that you've done that, you're logged in, you're ready to go with the app on your smartphone or on your tablet. These are the different species that you can report. Obviously, I'm not showing all of them. It's just showing you the different, taxonomic groups, essentially. You've got your diseases, you've got your animals, you've got plants, you've got insects. So obviously, in this case, you're going to go to plants, even though, it's a macroalgae. It just slots under plants. So you're going to go there, and then you're going to submit a new sighting. Now, in this case, this walkthrough, it is for Chinese mystery snail. So you're just going to do the exact same thing, but for Starry Stonewort. So again, this is your homepage, right? So you're obviously going to click on there for a new sighting. But what's really important here, and in speaking with Carol, it's important data, is the negative survey, right? So let's say you guys are out, you toss a rake, you pull it back, and you think that you didn't find any Starry Stonewort, you can actually submit a negative survey. So what that means is we'll get that report, but it'll just show that you didn't find it there, which is important information to have. And then there's your upload queue, which I'll come back to. And then, your groups of plants and animals and various groups. So we're going to click on new sighting. So this is where we're going to submit a new sighting of Starry Stonewort. So let's say you're out in the field, you think you found it after doing a rake toss, you click on new sighting, it's going to open up. You're going to click on plants. In this case, I apologize, but it's going to say wildlife because Chinese mystery snail. So you're going to click on plants, and then you're going to navigate to Starry Stonewort. You can either type it in or just scroll through. It's usually much easier just to type it in at the top. And then you're going to select Starry Stonewort, obviously. And then it's going to bring you to this page. It's going to auto-populate the species that you're reporting. And remember before how we talked about allowing the app to track you? It's going to give you the latitude and longitude. Now, this accuracy is not very good because when I did this, I was like in my basement, basically a bomb shelter. So I don't think it was able to pick it up very well. But usually the accuracy is much better than this, especially if you're out, on the countryside. So it's going to auto-populate lat long, and it's going to populate the species. So the only thing you have to do is tap here to add a photo, right? then it's gonna ask you for access to your phone's camera, which you're just gonna say yes, and then you're gonna snap a photo of the species, and then you're gonna save that report. Okay, because now you've got the location, you've got the species, you've got, you know, the dates already gonna be plugged in, and yeah, so you basically got all the information that we require on our end. And once you click save, it's gonna go into your queue, right? So then that queue that we talked about before, all you gotta do is go in there, you're gonna click on it, and at the end of the day, you might have 50, you might have 5, you might have 1, and you're gonna see them all here. Then you're gonna click on actions, and then it's going to pop up like little, you know, check boxes. You can check them all. And then once you're back on Wi-Fi, obviously, and then click upload. So then when you click upload, all those reports are going to come to us. So big takeaways here. Make sure you're on Wi-Fi when you're uploading reports, and make sure you log in. If you don't have a login, make sure you create one with an email and your name. And then honestly, at that point, you guys are good to start reporting. So your report is up, you know, it'll give you a notification saying that it's been sent to us. You'll also receive an email when your report has been verified, or it might even be deleted. Like if it's the wrong species, we'll click, you know, we'll delete it, but we'll submit a comment to you telling you what it was. So that's the only time you'll receive emails from us, is when we're either asking for additional information like photos, or if, you know, your report has been verified and released, or potentially deleted because it's the wrong species. Okay. So that being said, that was the app. You know, it is fairly intuitive, and I'm not going to lie, way better than the EDDMapS Ontario app that we had. You know, we used that for years, and I even disliked it. Even to this day, I was like, I don't know how to do this, because it wasn't very intuitive, but that one's very basic. You know, report positive sightings. You can create a negative survey. Again, that's when you don't find what you're looking for. And you can, you know, you just have your upload queue. So it's really basic, bare bones stuff. Easy to use as long as you have the tools to do so. So I know that we're running out of time here, but I am going to walk you through here. Please ignore all of my things that I have open. Okay, so this is the webpage, right? And this is eddmaps.org. Okay, so just when you're going to the browser, www.eddmaps.org, it's going to bring you here, right? You've got all sorts of cool tools here. There's tools and training for doing data downloads. There's species information, which, by the way, finally has starry stonewort information. I was fighting with them for a number of, well, what seems like years to get it uploaded. And I had them on the phone the other day, and I asked them to do it while I was watching. So they finally did it. So you have, you know, lots of information here. I am going to get some additional photos added to this. So Chuck at the University of Georgia asked Andrea and Tyler to provide some more photos. So I'm going to be doing that. And then you can also look up distribution maps, right? So real quick, let's do that. Sorry. So you can see there's 4,400 reports like globally, but in Ontario, we have, these are all the reports that have been submitted to us, mainly as bulk uploads. And Tyler, I would actually encourage you, if you do have some time, you can go through some of those to make sure that they are what they are. But, you know, as members of the public, you guys can look this up, right? Like you can go to all these individual reports, and as you zoom in, they obviously spatially spread out. So you can see here in Stony Lake, I've got the data in here that was submitted by Brian Ginn at the Lake Simcoe Region Conservation Authority. So there's all sorts of sightings. Yeah, like, you know, Tyler said, all the way over to Kingston. Okay. So that's distribution maps, species information, but here's the important part, right? You guys want to report sightings. So if you can't do it on a phone, this is how you're going to do it. But you're gonna have to make sure that you have something to take a photo with, right, while you're out in the field. So whether it's a camera, or, you know, you have a friend who has a smartphone or something along those lines. And then what you're gonna do is you're gonna go to plants, right? Now it's gonna give you all the jurisdictions that have EDDMapS. So you're gonna look here, Ontario. It's gonna bring up a very similar reporting page that you'd see on the app. Now it's asking for species. Now what you're gonna see here is all the red, as it says right here, are required, right? So in the app, remember how it's like really bare bones? In here, you can actually add a lot more information, but what's only required is in red. So if you don't have other information, don't worry about it. Just submit the most important stuff, which is obviously species. So we're gonna go starry stonewort, right? We're gonna say it was, yep, today's date. We're gonna say infested area seemed like it was 10 square feet. And habitat, we're gonna say it was at the edge of a lake, right? Just some additional stuff. You could do, it was a dense monoculture, which it usually seems to be. I don't know. If, yeah. And then you can even put like plant description, whether you know, mature or in flower, in fruit, if you see the bulbils. And then you're gonna have to plug in the lat long, right? So this is where some people might get hung up. They're like, I don't know how to find out lat long. All I do when I want to find out lat long, and I'll do it real quick here for you, is I go to Google Maps. Okay, I type in Google Maps in my browser. I go here, and then you're just gonna, I don't know why it took me there. So you're gonna zoom into your location, right? You're like, oh, I was right here looking for at Starry Stonewort on Pigeon Lake. You're gonna click to the nearest location that you can get, and you see down here this little dialog box. It's got your lat long. I always just highlight it, I copy it, and then I just throw it in here. So that's how I do it. I mean, there's multiple ways of getting lat long, but I just find that the easiest, just going to Google Maps, zooming into like the closest geographic spot, and then copying and pasting it. Because then when you do that, you're going to say place marker at my position, and look, it's right exactly where I clicked on Google Maps, right? You can also do various things here. You can do a point, like if you only found, one little plant. If you think it's a huge area, you can actually draw a polygon. So you can just zoom into your map here, and you can actually draw like the area that you think that it was inhabiting. And we'll see that when you report that to us. You can also add some description, like if there's some things that you want to know, like, oh, I was found in amongst, Eurasian water milfoil and Canadian Elodia. Feel free to throw that information in there. Right here is where you're going to upload your photos. My recommendation to you is to save the photos that you have to your desktop. It's usually the easiest place to find it, or put them in a file on your desktop, because then you're going to choose file. And then what you're going to do is you're going to upload the photo from your desktop, and it's going to come to us, right, for review. And then you can add more comments if you want and submit report. Okay. So when you submit that, again, it's coming to us, and we're going to verify it, and we'll release it. And you'll get the same email notifications and all that like you would if you're using the app. Okay. So that's really the nuts and bolts. It's, it might seem complicated, but it's really easy. It is really intuitive. Like once you get in there, you make a profile, you're good to go. That being said, if you guys have any questions, I'd be more than happy to answer them now. Unfortunately, I don't have a slide with my email on it, so I'll throw it in the chat box afterwards. So if you guys have any questions about EDDMapS, you can just email me directly. But yeah, in the meantime, so you have the phone, right? 1-800-563-7711. You can call us. I'll answer it, or somebody else will answer it. If we don't answer it, leave us a voicemail. You've got EDDMapS application. You've got the EDDMapS web browser. And, most importantly, you have starrystonewort@gmail.com, which will go directly to Tyler. So you have a plethora of means with which you can report to get that information into us. So hopefully that helps.
Thank you, Brooke. That was fantastic. Would you be willing to share this presentation with me, and I'll get that out into the resource?
Oh yeah, definitely.
So you'll have this video. We'll have a video of this entire day's presentation up within a day, and we'll, in the same list of resources, we'll put this presentation with I'll probably save the PDF format along with mine. And Tyler, if you don't mind, if you're willing to share yours as well, put them all together so you have all this information. Go back in your leisure and look at all those steps, how to do EDDMapS, or you can watch the video again. So we're pretty much at the end. We're a little afternoon, but we did okay. Any questions about anything? I know we've done an awful lot of information today. You guys are great for hanging in so long. I think you've got plenty of information at your fingertips. It might be a little overwhelming if you're completely new to this, but again, we're trying to make it so you can wade through these presentations, or watch the video again, or look at your the various fact sheets and guides we have made available. And again, I suggested with July through October. If you want to go out this afternoon and start throwing rakes, that's fantastic. You rake toss this afternoon, tomorrow, next week, whatever. It's okay. You can do them whenever you can, as often as you want. But the suggestions we gave earlier is kind of minimum we'd like to see. But that's what we'd like to do. We still got lots of conversation going back and forth through the chat box, which is fantastic. That's what this is all about. This is a collaborative, and you guys are collaborating. I want to thank Brooke and Tyler again too, and particularly for their, Brooke's work. As I mentioned, he and I've talked a fair amount about how to do this at EDDMapS and getting you guys into this, and then having Tyler and his folks there doing the confirmation. Shelby, I guess I never met you, Shelby, but I'm glad you're on board here. They're doing all the confirmations. It's just fantastic. Again, it goes back to that map I put in that network that you have built here in Ontario. It's just great. I mean, having those kind of resources to help you through this and confirm this is what it's all about. Somebody answered, I asked to have an account with EDDMapS. Ontario will work on EDDMapS. I think you said it would. Oh, Brooke just responded, okay. So I think we're about done, folks. It was a good session. I hope it wasn't too much information, but I think you're going to do fine. My experience, the work that citizen scientists have done is just excellent. It's all fantastic, good data. And you guys are really passionate about this, and that's what counts. And it's fun once you get out there throwing rakes and investigating what you've got. It's actually a lot of fun. I enjoy it. So last chance here for some questions. I will again save this chat and we'll go through it again and make sure there's any unanswered questions. We'll try and answer them. Presentation, we'll get it. Mike's in charge of that, but he can get it done probably by today, if not tomorrow. So it'll get up quickly. Brooke, I think there's a question to you. If you type in the KC project, is not on the list. I'm not sure.
Yeah, I think Jill, so one of the credentials that you can put in when registering is user group. I think that's where she's getting a little bit hung up. And Jill, I would just encourage you to just leave that blank. That's not something that you have to enter. Again, just make sure that you put your name, your first name, your last name, and your email address, and then a password.
And I will follow this up, if not today, tomorrow, with an email reiterating much of the contact information for all of us, and also where to find resources again. So we're going to hit you with that redundantly, but that way you'll be able to pick and choose what resources work best for you to identify these things and move forward. So with that, I think we're done. I don't see any more questions. Thank you all. This is a great session, and good luck out there in the field. I think you're going to have fun with it, and we're looking forward to the data you collect. Thank you, everybody, Brooke, Tyler, all the help we've gotten, all of you citizen scientists. It's just amazing how many people you got going.
I'd like to say thanks to you, David, for the fact that you're in New York, and yet you've helped Ontario so much. Establish this network and generate education and outreach for all of us. So just big props to you for all the work that you do and assisting Ontario as much as you have.
Well, we appreciate it, and again, going into this when we started the collaborative, I didn't realize there's no way to know that there'd be such a groundswell of people that wanted to get involved in Ontario, so we ran with it. We had a small group in Indiana last year. We had you guys last year. Now this year, growing dramatically. Our New York State work is growing rapidly, so it's all good. And again, we got people talking to folks in the Midwest who are doing research, and it's all about collaborating. So keep up the good work, all of you. All right, thank you, everybody. Have a great day.
Thanks again. Bye, everybody.